Hi there, and welcome to this video where we're going to be looking at a practice paper that I've made for my students. So if you're in my class, you've already done this, and if you're not in my class, you might want to do the paper. If you follow the link below in the description, you'll find this paper on my Google Drive. So this paper is a diversity and unified paper. At my school, we didn't have enough time basically to do 3 separate papers as mocks, so we did a combined paper which was in the style of unified, so that means longer questions, more 6 markers, more questions that are about analysis and evaluation. But this paper tested content that you might expect to find in diversity papers, biological diversity papers, or unified biology papers. So do this first and then come back to this paper to mark it. Now the mark breakdown of the paper is a bit complicated, and here's a PowerPoint slide which you can take a screen grab of if you want, which shows how many marks are in the paper and sort of how they link to the areas in the advanced information that we know is coming up. So there's a lot of marks in this paper about cloning and biotech and to do with practicals in cloning and biotech. There's quite a bit of marks in biomolecules, biodiversity. I struggled to find past questions on cellular control, but I really think the stuff on the Lac Operon might come up in your actual exams coming up in a few weeks' time. And then there's the rest of the marks available. You can pause and have a look. So if you've done the paper, let's get going. And if not, I recommend do the paper and then come back to this video for the full run through. All right, so let's get into the first question. The first question is about muscles. Now, I think this is a big prediction of mine for what might come up in the summer, because muscles really have never been tested as far as I can see. I've looked at the past papers across the last 4 years, and muscles have never come up. So this question gives you a picture of the proteins within the muscle fiber, within the sarcomere. So first of all, let's just kind of label these. A is actin. C is troponin, and B is tropomyosin, and then myosin is already labeled for us down here. So myosin binds to actin in the Crossbridge cycle, and that generates the force for muscles. And we can expect that potentially there's also something in this question about how calcium is involved with regulating this process, because we've got troponin and tropomyosin there. So let's look at the question. As the proteins in skeletal muscle myofibrils are arranged in groups to form a repeating pattern, as the muscle contracts, the pattern changes. Describe how the pattern within a myofibril changes when the skeletal muscle contracts. So this is all about the banding pattern. So I'm going to show you an animation about this, and then we're going to look at the answer. So if you didn't get this right, this animation is for you. This shows us a diagram of a muscle, of a sarcomere, of a muscle fiber, when viewed down a light microscope. And we see this characteristic banding pattern. We've got a lightly staining I band, a darker A band, and a kind of middle purple H band. So what are these bands? Or how do these bands change as the sarcomere contracts? Okay, this is the image that you need to remember. So the A band actually stays the same size. So the A band is this whole purple band here that I'm highlighting. Okay, so as the sarcomere fibers slide past each other, as the Crossbridge cycle moves along, the actin and myosin fibers slide past each other, called the power stroke, and the A band stays the same size, okay? Because the A band is really the total length of the myosin, okay? Pretty much. If I go back, you'll see what I mean. So the A band, if I go back, is basically the length of the myosin proteins. Now, there is a little zone within the A band called the H zone. So the H zone is part of the A band, and the H zone shrinks because the H zone is basically the part of the muscle fiber that has myosin, but where there isn't any actin. So as the 2 actin fibers at either end of the sarcomere move closer, the H zone is shrinking and shrinking with another power stroke, and shrinking, and it will actually disappear. Okay, so the A band stays the same size, but the H zone and the I band both shrink almost to nothing. So the I band, let's just take it back again, the I band is that part of the sarcomere where there isn't any myosin. So there's just actin and no myosin. So you can see this I band, if we go back another step to here, and we sort of step through, with each power stroke, the I band gets a little bit smaller and a little bit smaller until it goes almost to nothing. And then when the muscle relaxes, this banding pattern changes again. So the entire cycle repeats, and there we can see once again H and I zones or bands shrinking, and the A band stays the same size. Right, so let's go back to the question and look at the mark scheme. The mark scheme right here, the I band and H zone decrease or get shorter is one mark, and then the A band stays the same size or length is another mark. Other things that we could say, which I forgot to say in the little run through there, is that the Z lines, which are the two ends of the sarcomere, move closer together as the sarcomere shortens. But you needed that word. The thing that was tricky about this was you needed the word sarcomere, and you needed to talk about both the I band and the H zone decreasing. Not just one, but both. I band and H zone decreasing. So this is an old style question taken from an old paper, so they don't give a QWC mark anymore, but basically in your specification you always need to use this terminology well all the time. Basically this new specification is harder than the old one, so you need to be using these words. Okay, let's move on. So the next question is about how calcium regulates this process, and it talks about dystrophin. So this is one of those application questions where you get some new information. It says dystrophin is a protein that protects the membranes such as the sarcoplasmic reticulum. The sarcoplasmic reticulum stores calcium ions in the relaxed muscle cells. So this is telling you this, but you should actually know this already for your spec. You know that sarcoplasmic reticulum is where the calcium ions are held, and then calcium is released from there to initiate muscle contraction. So an early sign of DMD in muscles is a delay in muscle relaxation after a contraction. So this is a bit weird. Muscle relaxation after a contraction suggests why muscle cells fail to relax following a contraction if dystrophin is not functioning correctly or is absent. So this is kind of weird because you might expect that DMD affects the initiation of contraction, but it also affects the stopping of it. So let's look at another quick animation. So here's how calcium triggers the Crossbridge cycle. So before the muscle contracts, there is not a lot of calcium in the muscle cell. So what this means is that the myosin head is blocked from binding to the actin fiber because this troponin and tropomyosin are blocking the binding sites for the myosin head. Now, nerve impulses stimulate the release of calcium ions from the sarcoplasmic reticulum, and this calcium then binds to troponin, and it causes a conformational change such that tropomyosin shifts and moves away from the binding sites. So now, the activated myosin heads can attach to the actin, and as soon as they attach, they get this power stroke, and they pull along. Now, we'll go through this whole process because I really think this is coming up this year. So now they've moved along, and in order to release, ATP needs to bind. So ATP is actually needed for the head to detach. And as soon as ATP binds to that myosin head, it's hydrolyzed, and the energy is used to kind of spring the head back to the ready position, to its original state. And then it can bind again, and it levers along. ATP is used to kind of provide energy to spring it back to the ready state, and it keeps going and it keeps going. This is called the Crossbridge cycle. Now, for the muscle to stop contracting, we have to remove calcium from the sort of cytoplasm of the muscle cell, and that is done by actively transporting it back into the sarcoplasmic reticulum. Now, if the sarcoplasmic reticulum membrane is messed up, then this process, it won't be as quick and will actually be kind of slower because maybe some calcium leaks back out. So that is what the question was about. Let's go back to the question. So suggest why muscle cells fail to relax following a contraction. So let's just go straight to the mark scheme. The mark scheme says, well, calcium ions, or Ca2+, it would be better, are not reabsorbed. So the calcium ions, they stay bound to troponin, which means that tropomyosin will still be shifted away from the binding sites. So the binding sites are still exposed, which means cross bridges keep forming, or you could have said myosin stays bound to actin and continues to move along the actin fiber. Okay, so this is really what you needed to talk about, that specific detail about how calcium regulates the cross-bridge cycle. Okay, if you want to have a look at the examiner's comments there, please pause and be my guest. Now, the final part of this question is about an inherited disease, the same one we just talked about, DMD, and it says it's caused by a mutation in a gene that codes for a protein called dystrophin. Dystrophin is present and functioning in normal muscle cells, and it's asking you to explain how a gene mutation can lead to a protein malfunctioning. So again, this is taken from an Olsbeck paper. So the marks here are mainly for kind of sort of talking about how protein structure is coded for. But gene mutations is again something that I think might come up this year. So perhaps this year there might be even maybe a 6-mark question, which is a little bit about point mutations and different types of point mutation, and a little bit about protein structure. So I want to show you an image. So I've added this image over here on the right-hand side to kind of remind ourselves about this overall process and sort of think where the marks are. Okay, so first thing, if we have a gene mutation, we have a change in the DNA. Okay, so change in here, and maybe there's marks for saying there's a change in a specific base. Maybe that can then change a triplet. Okay, a different triplet would lead to a different codon, different codon on the messenger RNA. And if there's a different codon on the messenger RNA, then a different tRNA will bind, bringing a different amino acid here, which means that once we've made it into a protein, there'll be a different amino acid, and when that protein coils up in its 3D shape, it will coil differently because there maybe the amino acid is hydrophobic instead of hydrophilic. It just has a different charge, a different shape, and that affects the bonding in the protein. Okay, so there's lots of things you could have said here. Let's look at the mark scheme. So, first of all, idea that there's a change in the DNA triplet, or the code. Change in the codon, that's a key word, in the messenger RNA codon, which would give a different tRNA or anticodon. The anticodon is the 3 letters on the tRNA that will bind to the codon. So different amino acid, or you could have said different primary structure. I should have mentioned R groups when I went through that just then. There was marks available for talking about the different R groups, which have a different shape and charge. So there's different bonding. And this idea that the change leads to the protein changing its shape. So we could have talked about the hydrophobic and hydrophilic interactions and that kind of stuff. And then there's another mark for just saying that if the protein is kind of the wrong shape, then perhaps if it's an enzyme, its active site would be the wrong shape or something like that. Okay, so those are all the marks available there. There are lots of marks. I'll just go back to there so you can see them all. And if you need to have a little look, pause and have a look for yourself. Okay, on to the next question, question 2. So this question is basically about serial dilutions and aseptic technique, which again I think is a really key area that might come up. Okay, on to question 2. This question is about serial dilutions and working aseptically and everything to do with bacterial growth. All of these things, I think, are very likely to come up this year in your papers. Okay, so the question says, 2 students investigated the growth of bacteria at different temperatures. So different temperatures, that is our independent variable. 3 flasks containing identical solutions of nutrient broth were used. Flask 1 has the bacterium at 20. Flask 2 has the bacterium at 30. And flask 3 have no bacteria. So flask 3 is kind of a control, we can see. We've got 20 degrees C, 30 degrees C, and then 30 degrees C, but with no bacteria, so that's a control. Aseptic techniques were used throughout. Over the next 3 days, the students removed samples and examined the number of viable bacteria present. So that viable just means like living. Are they alive? State one further variable the student should have controlled in their investigation in order to produce valid results. It says further variable. So we can't talk about anything that's already been controlled. We've already controlled a bunch of things. We've already used 1 centimeter of broth, so we're not going to talk about that. We're using the same bacteria. We're certainly not talking about temperature, because that's the independent variable. So what other things can we talk about? Well, it's sometimes a bit tricky to think of these things on the fly, but you're talking about bacteria growing in a little jar. So if you struggle, always kind of think, try and imagine the experiment in your head. Talking about bacteria growing in a little jar, a little tiny flask, what things might affect the growth of that bacteria in the flask? Well, it's not nutrients and temperature, but what else? Well, bacteria need nutrients, but they also need oxygen. So maybe the oxygen level, maybe the pH of the broth might affect it. I suppose the exact same glassware, whether it's in light or dark, but these are kind of less likely. Probably oxygen and pH are your best bets. Let's look at the marked scheme. The volume of broth in the flask. So 1 centimeter of the bacteria broth was added, but we don't know the volume of the broth in the flask, the pH of the broth, and the oxygen concentration of the broth. All these things up here are your most likely answers. And here are some other things. So this is an interesting one, stirring or mixing before you withdraw the sample. If you've got your master culture beaker like this, and there's bacteria in it, a lot of the bacteria will settle to the bottom. So every time you take out the sample to test, you should mix it about to get an accurate reading, because otherwise if you take it from the top one time and the bottom another time, you'll get big differences. So those are the mark points available. Okay, so what do the students do? The students use the following procedure to determine the number of viable bacteria in each flask at a given time. From each flask, 0.1 centimeter cubed was removed and mixed with 9.9 centimeters of sterile water in a test tube. This tube was labeled A, and then the dilution goes as follows. Right, okay, so this is a bit baffling. From each tube A to F, 0.1 centimeter cube was cultured, and then the results are shown here. And use tube F to calculate the number of viable bacteria present in the original 0.1 sample. So tube F here has 1, 2, 3, 4, 5, 6 bacteria. And this is a serial dilution maths thing. So let's go back up here, because we actually need to know what was the dilution of each of these tubes. And it's misleading because it doesn't give you the information about tube A. Now tube A, remember, it was only 0.1 of the original flask and 9.9 of sterile water. So this is not a 10 times dilution, but this is a 100 times dilution. So A is already 10 to the minus 2 diluted. So that means that B has this plus 9, so this is 10 to the minus 3. C is 10 to the minus 4. D is 10 to the minus 5. E is 10 to the minus 6. And F is 10 to the minus 7. So tube F is a 10 to the minus 7 dilution. And that's the one we're looking at. This is a 10 to the minus 7 dilution. Okay, so there were 6 bacteria in the 10 to the minus 7 dilution. And remember that what was put onto this plate here was 0.1 centimeters cubed. It says it right here. 0.1 centimeter was cultured on the nutrient agar. And it's asking us how many bacteria were in 0.1 centimeters cubed. So there's no extra mass to do with that volume that was added. So we just need to basically take 6 and times it by 10 to the 7 because it was a 10 to the 7 dilution. So the answer is 6 times 10 to the 7 bacteria. Okay, and that's your answer. The mark scheme, 2 marks for that. 6 times 10 to the 7, 6 or 6.0 times 10 to the 7. And if you were just 1 out, you could have got a mark. So if you said 6 times 10 to the 8 or 6 times 10 to the 6, you could have got a mark. And basically it was quite tricky. There was the trick in there, which was really horrible for them to do, was this first bit up here. It was basically, if you didn't spot this, the fact that A was a 10 to the minus 2 dilution already, then you would have gone wrong. Okay, hard stuff there. Okay, the students disagreed about which tube's results to use as a starting point for their calculation. Discuss whether the Petri dish resulting from tube F was the most appropriate for them to use. Well, it's interesting here. Tube F had 6 bacteria, and it's easy to count. Now, tube E, you can also count the number of bacteria. 1 second. Okay, so in tube E, if you count it, there's actually 35 colonies there. It's a bit of a pain to count them all, but you can count them. There's 35 bacteria there. So now this would be a 10 to the minus 6 dilution here. So if you were to work this out, if you were to use this as opposed to this figure and estimate from here, you would actually get 35 times 10 to the 6, or 3.5 times 10 to the 7. So those numbers are quite different, aren't they? This would be the estimate using tube E. This would be the estimate using tube F. So which is more accurate? Which is more reliable? Well, the answer is actually E, because any anomalies would have a smaller impact. So you might expect, if you repeated this experiment, sometimes you get 35 colonies, sometimes you get maybe 30 colonies, sometimes you get maybe 38 colonies. They would vary a little bit. But all those numbers, if you plug them into the maths, you get sort of 3 times 10 to the 7, 3.5 times 10 to the 7, or 3.8 times 10 to the 7. So the effect of the anomalies would be reduced. Over here in tube F, well, maybe there have been some anomalies here. Instead of maybe there are only 3 bacteria coming from the actual solution that you've put on. Let's just say these 3, 1, 2, and 3, and maybe these other 3 bacteria are from contamination. So any anomalies from contamination would have a big impact. Let's say if those 3 bacteria there were from contamination, well, that's actually doubled our estimate. So the answer is that actually F would be better. So E would be better, my bad. So how do you explain that? Well, 1 mark for just saying we should have used E. It has the more colonies. So you always want to use the plate that has the most colonies that are realistically countable. So if it's got like, 200 colonies, realistically maybe it's not countable. But if it's, somewhere below 50, I'd say, that's kind of countable. And the reason is that the anomalies will have a smaller effect. And anomalies, mainly in this sort of thing, anomalies would be coming from contamination with other bacteria. And so actually, I think that it's not on the mark scheme, but I think this gives you a more precise, also a more precise answer, because you could get something like 3.5 times 10 to the 7 as opposed to 6 times 10 to the 7. It's more precise because you can give it to higher decimal places as well. So, but that was not on the mark scheme. There's some other mark points, and if you want to look at that, you can pause here and look at the examiner's comments. All right, on to this next question, which was a 6 marker and was quite challenging. So this question says, the process results from the student's investigation as shown in table 19.2. Now we've got information here about how the bacteria grew in different temperatures. We've got 20 degrees C and we've got the 30 degrees C. Now the information is given as standard form, so there were some problems, some students found problems kind of like analysing the standard form and recognising when things were going up and when things were going down. And the other problem with the 6 marker was you had to compare this to your standard kind of bacterial growth curve and use the correct terminology. So I've prepared a little picture here, which I've just dropped in there, because I want us to think about the different phases of bacterial growth and try and work out where we are on this growth curve with the different kind of temperatures. So let's color code, okay? So lag phase, let's do as sort of pink, okay? So that's where the bacteria are kind of not really growing. And we could basically say that in 20 degrees C here, we're in lag phase because, in fact, the population goes down a teeny bit. I'm just going to zoom in a little bit more and move across here. Yeah. Whereas at 30 degrees C, well, no, actually, it's a similar lag phase here. At 30 degrees C, you know, there's not much change. 7.1, 7.4, not really doing much. Right, now let's talk about the exponential phase. So this is where we would expect the log of the numbers of bacteria to be increasing, you know, quite steadily. So we'd expect the powers of 10 to be going up, 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 up. So where do we have that? Well, this is going 4, 7, 9, 10, 7.8, and then we max out here. So the log phase is kind of there, 4, 7, 9, 10, looking at the logs, the powers of 10. Over here, we've got 6, and then 10, and then 11, when it's still growing. Stationary phase, let's color code that blue here. So stationary phase is, you know, just when it's peaking. And I would maybe say that stationary phase is about there. It's starting to decline, isn't it, really? So it's kind of stationary / death phase there. Maybe we just say, let's actually just say that the stationary phase is at the max point there. Stationary phase here, the max point is, well, we can say, I guess, there. 12 and 18 hours, it's at stationary phase. And then we're getting into death phase. Let's give death phase, let's make it green. So death phase is when the numbers are dropping. So death phase is kind of here and over here. Okay, so let's label those again. We've got the lag phase, and we could say that the lag phase for both 20 degrees C and 30 degrees C is similar. It's the same duration. Then the exponential phase. The exponential phase is quicker in 30 degrees C than it is at 20 degrees C. So why is this? Well, bacteria can replicate faster at higher temperatures. Higher temperatures, 30 degrees C, closer to the optimum temperature for all the bacterial enzymes, means that metabolic reactions can occur more quickly, respiration can occur more quickly, and bacteria can replicate more quickly. So that's why that's the explanation for the differences in exponential phase. Well, exponential phase finishes more quickly at higher temperatures because the bacteria have grown more quickly and have kind of used up resources. Remember, stationary phase is when resources are becoming limiting. So because the bacteria are growing more quickly at 30 degrees C, they reach stationary phase more quickly. They reach stationary phase at 18 hours, whereas in the 20 degrees, sorry, my bad, not 18 hours. They reach stationary phase at, I meant to do this one, isn't it? Like 12 to 18 hours, 12 to 18 hours. Whereas in the 20 degrees C, it's 24 hours. So quoting data. And then the bacteria enter death phase or decline phase sooner at 30 degrees C. At 24 hours, we're already in the death phase. The numbers are going down. Whereas it's 36 hours for the 20 degrees C. The numbers are going down. Okay? So that's everything you needed to say. How do examiners mark this sort of question? Well, here we go. Let's start with 1 or 2 marks, okay? 1 or 2 marks down here. Describe some differences between the 2 temperatures. Or explains a reason, okay? Gives an explanation. But, this is the thing, not enough technical terms, okay? The communication is hampered by inappropriate use or not enough use of technical terms. So what are we talking about technical terms? Lag, log or exponential, stationary, and death. You've got to use those terms. If you didn't, you could only get 1 or 2 marks. The next level up here, level 3 or 4 marks, describes some of the differences, some use of key terms, but not all of them, and you've got to explain a difference. So maybe explaining that the bacteria growing at 30 degrees C reach stationary phase sooner because the nutrients run out. Whereas in the other one, it takes longer because the nutrients are basically around for longer because the bacteria take longer to grow. And then for the top level, level 5 or 6, describe the main differences between the 2 temperatures using the key terms. So you've got to talk about each phase, really, lag, log, stationary, and death, and explain them and use the key terms. So all the kind of points that you could have said are here, the comparison of the curves, and the explanation here. So pause there if you'd like to have a bit more of a look. Just before we move on, I've just added an exemplar. I found this online. So this is a 6-mark answer. This is published from the examiner's report. The writing's a bit tricky to read, but this is a real student answer from 2018 when this question came up. And this was given 6 marks. So if you want to read it, there you go. Okay, following on from that question, part 2, no bacteria detected at any time in the flask that was inoculated with nutrient broth that did not have bacteria. Explain the purpose of the flask. Well, basically this is control, and it's to check for contamination. So it's really to check whether the aseptic technique was carried out correctly, because if the student didn't do aseptic technique correctly, you probably expect some bacteria would end up in that broth anyway. So that's the reason. I'll show you the mark scheme in just a second. Part 3, the teacher said they shouldn't grow the bacteria at 35. Well, 35 is close to 37. And 37 degrees C is body temperature. So body temperature is, if you culture bacteria at body temperature, you're likely to culture pathogens. And then finally, before I show you the mark scheme for these extra questions, the student should have carried out the investigation using 3 flasks at each temperature. How would this have improved the student's investigation? Well, remember we talked about anomalies before. Well, if you use 3 flasks, the effect of anomalies is reduced because they average out. So if you use 3 flasks, you can get 3 readings for your bacterial population size, and then add them together, average them out to get a mean. And this would increase accuracy and reliability. So let's look at the mark scheme for those 3 little questions that I blitzed through just then. So the first about the flask is a control and the idea of checking for contamination. Control and contamination. The second one, it just could encourage the growth of pathogens. I wonder if you need to say human pathogens. Probably a good idea to say human pathogens, not just pathogens. Probably pathogens would be fine, I'd imagine. And then the next one, reduced impact of anomalous results. Increases repeatability. It says allow reliability, but I really should have said repeatability there. And calculated mean would be more accurate, is I think what I said. Calculated mean is likely to be more accurate, and that's because random errors average out. So therefore you get closer and closer to your true value, which is the actual bacterial size of the population. Okay? Okay, on to question 3. So the question says, bread contains a mixture of polypeptides known as gluten. Now, here's a little extra image just to illustrate that for you. Gluten consists of 2 types of polypeptide, gliadins and glutenins. So here's the structure of the gliadin, just to remind you a little bit about protein structure. And the glutenins and the gliadins kind of combine together in like a mesh to make gluten. Okay, so here's that picture. So gluten helps to trap the carbon dioxide within the bread dough because it's kind of like a stretchy network, and that means the bubbles of carbon dioxide produced by the yeast kind of get trapped and help the dough to rise. Just a little bit of baking science for you. So the species of yeast is called Saccharomyces cerevisiae, baker's yeast, and it's able to convert ethanol to acetyl-CoA at low glucose concentration. So it can, so we know that it can use glucose as a respiratory substrate. We know that, but it also can use ethanol as a respiratory substrate if there's not enough glucose. So figure 2 shows the oxygen consumption and carbon dioxide production of a population of yeast grown in batch culture. Now that is important, that batch culture phrase, because batch culture means nothing is added. Once the flask is sealed, everything's just contained. So nothing added, no nutrients are being added, and no waste products are being removed. Everything's just allowed to build up in the sealed container. So this question was kind of tricky, and the first reason that I think it was tricky is just there's a lot of information here. So I would always recommend taking in some highlighters to the exam, and like in a question like this, you know, for me, I am terrible at like processing black and white information. Like I just can't do it, so I'm going to color it in. So first of all, there's my oxygen consumption. Kind of starts really low, goes up, kind of plateaus, and then drops. There's my carbon dioxide production, goes really high, then drops to almost nothing, and then kind of gets going again at a lower level. Glucose concentration now in yellow starts high and then goes to 0, is already a 0 at about 9 minutes. And then finally there's ethanol, which kind of builds up until about 9 minutes, and then it kind of tapers off. Now, to kind of analyze this and figure out what's going on, you needed to remember something about yeast. So yeast is what's called a facultative anaerobe. So that means it can do anaerobic respiration or aerobic respiration. And weirdly, if there's a lot of sugar, it will actually do anaerobic respiration because it's faster than doing aerobic respiration. It is lazy. If there's lots of resources, it's just going to do anaerobic respiration, and it only really switches to aerobic respiration to get the maximum amount of energy if there's not enough glucose. So this will give you some help when looking at this question. So let's look at the different phases of the graph. The first, well, phases of this kind of process. The first phase is here, okay, at the start. At the start, which is from 0 to 9 minutes, what's going on? Glucose starts high and is broken down. So glucose is definitely being used as the respiratory substrate. Glucose is being broken down, and carbon dioxide is building up. What else is building up? Ethanol is building up, and the shape of the curve is almost exactly the same as the shape of the carbon dioxide curve. So this tells us that at the start, what's going on? Well, we're having glucose being fermented into ethanol plus CO2. Okay, so that's what's going on at the whole sort of start phase up here. Then, let's switch colors. Then, so the next phase is kind of here, I guess. Well, no, it goes all the way till here, really. And after 9 minutes, once the glucose is completely gone, something else has to be used to be respired. And now it's the ethanol. Remember the question just above, it says ethanol can be used as a respiratory substrate. There it is, ethanol to acetyl-CoA at low glucose concentrations. So have we got a low glucose concentration? Yes, it's gone to 0. So now the ethanol is being used. So now ethanol, you know, I guess the equation of this would be ethanol plus oxygen goes to CO2 plus water. Okay, because this is kind of aerobic respiration of the ethanol. And this makes sense because the oxygen consumption, which was low because it was anaerobic respiration, then starts to pick up. Oxygen consumption starts to pick up. We're switching, kind of this region here is kind of switching over to aerobic respiration. And look at the ethanol, it's going down, 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 until the ethanol is completely gone, and then there's no more oxygen consumption because there's nothing more to respire. And at this point, I guess the yeast would probably die. Okay, so that is the full analysis of this graph, which was really tricky. the key thing you need to remember is that yeast can do both aerobic and anaerobic. You need to know the equations for both. And you need to know that yeast actually will respire anaerobically, sometimes even if there is oxygen present. Okay, it can do it once because it's faster and it's lazy, basically. So let's look at the mark scheme. Here it is. So points given marks. Anaerobic respiration at the start. So just that would be sufficient. You could say glucose is converted into ethanol. Then respiration decreases rapidly or stops once glucose used up, and then it's ethanol. Ethanol is then used as the respiratory substrate or the carbon source once glucose has been consumed, and this is aerobic respiration because the oxygen demand goes up. And we didn't talk about this so much. Why is that? Well, it's because ethanol is fed into the Krebs cycle. And remember the Krebs cycle, let's just annotate here, the Krebs cycle produces NADH and FADH. And remember those go into the electron transport chain, and it's the electron transport chain that needs oxygen. It's the final electron acceptor. It's the only place we need oxygen, but because that ethanol is going into the Krebs cycle, we're going to need oxygen for it. And then finally, respiration fully stops once all the ethanol has been used up. Okay, and the examiner's comment there is interesting to read if you want to. Pause and have a read. It was a good discriminator. That means it was a tough question. All right, on to the next part of this question. So this is describe 2 practical considerations to ensure that the yeast population grows successfully once the initial culture is established. So remember that the question's already told us that this is batch culture. So there's lots of things you can say, but really the only thing you can't say is adding more nutrients, because that wouldn't be batch culture, that would be continuous culture. So I'll just show you the mark scheme, what's allowed. Aseptic techniques was a good thing to say. So remember, if you didn't use aseptic technique, then you'd have other microbes in the yeast population and it'd be contaminated. It might not work properly. And then you could have said a source of nutrients or respiratory substrates. You could have said a suitable temperature. This is probably a sensible thing to say. I probably would have said suitable temperature myself, and maybe aseptic techniques. You also could have got marks for saying use a pH buffer. Now, if you've forgotten what a buffer is a solution that maintains a pH. If you do chemistry A level as well, you might know that. It maintains a pH, doesn't, or it resists changes to pH, kind of moderates it. And you could have also said stirring or mixing, actually. It says accept mixing over here. So yeah, you might want to just double check on the right-hand side. There's lots of accept here. Accept a description and all these things here. So pause there if you'd like to have a look, and also just scroll down just a teeny bit if you want to look at the examiner's comment there as well. You can pause. All right, let's move on. Okay, here it says scientists wanted to estimate the number of yeast cells in a 25 centimeter solution of S. cerevisiae. They carried out the following 2 dilutions. 1 centimetre of the original solution was mixed with 9 centimetres of nutrient solution to make solution 2, so that's 10 to the minus 1. And then 1 centimetre solution 2 was mixed with 9 centimetres of nutrient solution to make solution 3, so that's 10 to the minus 2. The scientists then transferred 0.1 centimetre cube of solution 3 onto an agar plate, and 15 separate colonies grew on the plate. Okay, so what does this mean? So you've got to do a little bit of maths before you can get to the kind of dilution. So 15 colonies in 0.1 of solution 3. So therefore, you would expect 150 colonies, you might have expected, if it was 1 centimetre cubed of solution 3. So now we've got to the sort of solution 3 colonies per centimetre cubed figure. And remember, this was a 10 to the minus 2 dilution, or 100 dilution. So we'd have to times this number by 100, which would equal 150 hang on a minute, sorry, no, that's wrong. 15,000 colonies, I'll just write C for colonies, per milliliter of the original solution. Okay, but that's not quite what we need yet. That's per mil, but the question wants us to work out how many in 25 centimeter cubed. So now we need to times it by 25, and the answer you get is 375,000. But the question doesn't ask for it like that. It asks it for an answer in standard form to 3 significant figures. So 3 significant figures is easy because this kind of already is 3 significant figures, if you like. So it's 3.75. I remember when doing standard form, you kind of count how many positions you move the decimal place. So let's do that in blue here. So if it was 3.75, I'd need to times it by 1, 2, 3, 4, 5. 10 times 10 to the 5. So 3.75 times 10 to the 5 is your answer. And let's just look at the mark scheme super quick to see how many marks that is. That's 2 marks, 1 for the 3.75 and 1 for the 10 to the 5. So even if you got the standard form bit wrong, you might have still got a mark for 3.75. All right, moving on. Okay, now we're into 4. This is a question on sort of control of the heart rate in ECGs. Apologies, this question's kind of gone over 2 pages. It's a bit weird, but at the top here it says phenylchromocytoma. That's tricky to say. Pheochromocytoma is a rare tumour of the adrenal gland tissue. It can cause increased hormone release from the adrenal glands. And here's some ECG traces. X, normal heart rhythm. Y has this phenomycotroma. And Z has bradycardia. Now, you need to know what a normal one looks like. And you need to know that bradycardia is a slow heart rate. And then you should be able to make the link that adrenaline speeds up the heart rate. So this one's going to be fast. So which one's normal, which one's slow, and which one's fast? The normal one is this one. It's kind of the middle one. The slower one is the top one. And then the faster one is going to be this one down the bottom. So that would be Y. So Z, X, and Y. And that's for 2 marks. Yeah, Z then X for the first mark and Y for the second mark. Okay. Then it says sketch a trace for a patient who has entered atrial fibrillation. So atrial fibrillation is it's kind of like a mini heart attack, I guess. So not all of the heart is fibrillating, which is kind of what it is when it's a heart attack, but just the atria are. So the ventricles still have the normal contraction, but the atria sort of part of it is all messed up. So basically it looks like this. It's like a wiggly, wiggly, wiggly line with a big ventral spike and a wiggly, wiggly, wiggly line. And a ventricle spike again like this. So oops, wiggle, wiggle, wiggle. Oops, like that, and so on. Okay, so you get 2 marks for that. 1 for the fact that they're sort of clear spikes and 1 for the wiggles. Let's look at the mark scheme. It says large single peaks present, and then the small wavy line with at least 3 wiggles between any 2 peaks. So here's an example of what it would look like down here, which would get 2 marks. Next question. Suggest why reduced heart rate is sometimes seen in people who are aerobically fit. Okay, so so this question showed up some misconceptions that some students have. So some students think that if you're aerobically fit, you need less oxygen. Okay, that's not the case. It's not that you need less oxygen so your heart beats less. It's just that your heart can beat more effectively, probably because it maybe has a larger volume. So the heart actually increases with aerobic exercise. Its volume can slightly increase, and also the heart muscle might be able to contract with more force. All of these things mean that you have a greater cardiac output. So let's look at the mark scheme. So first thing to say is increased stroke volume. Remember, stroke volume is the total amount of fluid, or blood obviously, total amount of fluid that the heart can eject with 1 beat. So that's the AW, if you'd said that, that's kind of what it means. And that's because there's increased volume of the ventricle. And then you would have also got a mark for saying increased strength of heart muscle, or increased thickness of the heart muscle, or myocardium. So yeah, this is that misconception. The examiner, when this question was given to students in the actual real exam, there was this misconception that loads of students around the country had. Says there appeared to be a misconception amongst many candidates that aerobically fit people would not need as much oxygen delivered to cells, that they had more red blood cells or needed less ATP. So, I mean, aerobically fit people might have more red blood cells, but that wouldn't mean that their heart would beat slower. But they'd certainly need as much ATP to sort of power their muscles, and they need as much oxygen. It's just that the heart itself can change in structure. Okay, on to the next question, question 5. Hopefully you found this relatively easy. There's lots of easy marks here. It's just identify errors and correcting them. So you have to identify and correct an error for each mark here. So I've put in a picture here in this other version of the paper that I have, just to remind you of the kind of content that you needed to know that it's testing about, so you can kind of refer to this over here. So let's read through the thing. When the heart rate is too low, the level of carboxylic acid in the blood, there's no carboxylic acid in the blood, that's incorrect. It could be carbonic acid, is an acid that is present in the blood, which is kind of H2CO3, which then dissociates into hydrogen carbonate and H+ ions. What else? Let's pinch in a little bit. The vagus nerve sends axon potentials to the AVN to increase the contraction. Well, there's two mistakes there. The vagus nerve is not the one that speeds up the heart, and it doesn't send impulses to the AVN. Look here, it's the accelerans nerve, increases the heart rate, and it sends it to the SAN, the sinoatrial node. So that would be the correction for this point. The baroreceptors in the walls of the blood vessels then detect at the full of pH. No, that's not correct. Baroreceptors detect pressure receptors. So these stretch receptors here are, those are in muscles, my bad. It's these ones, the blood pressure directed by the carotid sinus. These are baroreceptors. Baroreceptors that are actually in the carotid sinus, which is kind of this region right about here. The blood vessels kind of merge at something called the carotid sinus there. Then it says the endocrine system can also change heart rate. Release of the hormone adrenaline from the adrenal medulla causes the smooth muscle of the heart, no thank you, it's not smooth muscle, it's cardiac muscle of the heart to contract more frequently. So those are the mistakes. We've kind of already corrected them, but let's just show you the mark scheme. So carboxylic should be carbonic, not carboxylic, or you could have said H2CO3. The vagus nerve should be the accelerans nerve, or you could have said accelerator, or you could have actually just mentioned that it's part of the sympathetic nervous system. The AVN should be the SAN instead. Baroreceptors should be pressure, or you could have kind of talked about pH receptors or chemoreceptors. So baroreceptors, pressure, or you could have talked about chemoreceptors if you wanted to stay with pH. And smooth muscle should be cardiac muscle. Okay. And if you weren't sure on that, you can just pause right there and try and memorize that diagram, because that's what this question was testing. All right, figure 20 shows the disaccharide lactose, which is found in milk. So this is lactose here. One of the monomers of lactose is galactose. Bacterium E. coli usually uses glucose as a respiratory substrate, but under certain circumstances, E. coli is able to use galactose as a respiratory substrate by breaking down lactose into glucose and galactose, and then using both glucose and galactose as respiratory substrates. So this says, explain how the structure of galactose allows it to be used as a respiratory substrate. Which is glucose, which is galactose here? I'm not so sure, to be honest. It's difficult for me to identify which is which. To be honest, I'm not even going to try it. Let's just say that one's glucose and one's galactose. They're really similar, aren't they? They're both hexose sugars. They both have lots of carbon-hydrogen bonds and carbon-oxygen bonds and oxygen-hydrogen bonds. So, you know, we can expect that you might be able to convert galactose to glucose quite quickly and easily by an isomerase enzyme. So isomerases can generally convert one hexose into another. And this question I took from an older specification, an older syllabus, so the mark scheme is a bit weird. So if you didn't do so well on this, I wouldn't worry too much. The mark scheme said what you could have been awarded for, that bonds contain chemical energy, can be broken by enzymes. So this is one tick, this is another tick. It's soluble, so it can move within the cell. And I would probably expand on this AVP, because this, again, I took this from an old syllabus. So I would expect that the AVP, you could get maybe 1, 2, or even 3 marks from the AVP. I'd accept used in glycolysis, or converted to glucose. And if you mentioned isomerase enzymes, I think I would also give that. Maybe even the fact that it's a hexose sugar might have been worthy of a mark. So it's a bit of a strange mark scheme because it's from an older specification. But I want to include this because it goes on to talk about this question. Goes on to talk about some stuff that I think really could be coming up. So now it says, E. coli usually grows in conditions where the extracellular concentration of lactose is low. That's interesting. I don't know why is it giving us that information. Extracellular concentration of lactose is low. Okay, so I'm already thinking from that sentence, I'm thinking about like concentration gradients, because it's told us that the concentration is low outside. In such conditions, lactose does not easily cross the bacterial cell surface membrane. Suggest and explain why lactose is unable to cross the membrane. Well, we can say a few things. First of all, from this point here, there's a low concentration gradient. So potentially the concentration is not high enough to sort of drive diffusion or transport via carrier protein. And it probably wouldn't be diffusion, because this other point you could say is the fact that this is a big molecule. It's a big molecule which is polar and it's too large to fit between the gaps between the phospholipids. Or you could say it's not lipid soluble, so it would not be able to cross the phospholipid bilayer. So the marks available. First of all, it's too big to pass between the phospholipids. Again, this is an older spec, so this is probably still worthy of credit in the new spec, but I would say in the new spec, you would probably get a mark for saying not lipid soluble. And sort of therefore can't dissolve in the bilayer, or the fatty acids part of the bilayer, just to really be sure. That would probably get you a mark. Oops, excuse my writing there, it's a bit messy. And potentially you could have got a mark for saying it needs a carrier protein and there's a small concentration gradient. That's what I said as well. That was the information that was given in the question. Okay, so those are 2 slightly weird questions with the old mark scheme, but the reason I put this in is because I wanted to put in a question about Lac Operon. And this question is about the Lac Operon. Many of my students didn't quite spot that, but I kind of have a feeling that Lac Operon could be coming up this year. So the question says, in order for lactose to enter the cytoplasm of E. coli, a protein is required. The E. coli living in the digestive system of young mammals are more likely to contain this protein than E. coli living in the digestive system of old mammals. Suggest an explanation for this observation. Well, young mammals, what do mammals do? They produce milk. So young mammals drink milk. Talking about baby mammals, okay, or recently weaned mammals that have kind of recently been drinking a lot of milk. So milk contains lactose, and lactose can turn on a gene. So this is about control of gene expression. So I'm just going to quickly show you the animation about the Lacquer Operon, if you didn't realise this or if you've forgotten it, and then we'll go back to the mark scheme. Okay, so here's E. coli. And bacteria like E. coli only produce proteins and enzymes to break down lactose if it's necessary. They won't do it if they know that there's no lactose around. So bacteria in the small intestine of young mammals are exposed to lactose, and they'll turn on the production of these two proteins. The first protein is called lactose permease. That is the protein that helps transport lactose into the cell. It is a transport protein or a carrier protein in the cell membrane. The second protein is beta-galactosidase, and that is an enzyme that is the one that can hydrolyze the disaccharide into two monosaccharides. So the genes for this for these proteins are contained together in one functional unit that's called an operon. So the Lac Operon contains a promoter, an operator, the two genes Z and Y, Z being the membrane protein and Y being the enzyme. And then there's also an I regulatory gene, which is further away. So how do these all work? Well, under normal circumstances, when lactose is not present, I will produce a protein. I is transcribed and translated, and a protein is made. This repressor protein can bind to DNA, but it only binds on the operator region. So when it binds on the operator region, it kind of covers the promoter region as well. So that means it's kind of masking the promoter. Now the promoter is where RNA polymerase would normally bind, RNA polymerase to do transcription. So if that protein is there, the RNA polymerase, it cannot bind, okay, it bounces off, right? So that means that the structural genes, Z and Y, can't be transcribed and turned into mRNA and then into proteins. So we don't express those two proteins. So this is the reason why old mammals who don't have a lot of milk, so the E. coli in the old mammals' intestines, who haven't been exposed to lactose, well, they don't produce the proteins Z and Y. They don't produce this membrane transport protein. But what about young mammals? Young mammals, where there is milk, there's lactose. Now lactose, I'll show you that again, actually binds to the inhibitor protein. That's this one here, or repressor protein, sorry. And when it binds to the repressor protein, there's a slight conformational change in shape. So the repressor protein falls off the operator. Now the operator is clear, and so is the promoter. So under this circumstance, when lactose is present, RNA polymerase binds to the promoter, and it causes transcription of those genes into messenger RNA, and that messenger RNA is then translated by ribosomes to produce the two proteins. Okay, so that's just a quick recap on the Lac Operon, and now let's go back to the mark scheme to see what you needed to say in this specific question. Now it's worth saying before I do that this specific question was a short question, but I feel like this might be a longer question. There might be more detail about the Lac Operon coming up this year. Okay, mark scheme. So the mammal diet has high lactose concentration. You could have just said that lactose is present in the intestines of young mammals. The structural gene, or so you should have said the structural gene, or the lactose permease gene, or LAC-Y is transcribed or expressed or switched on, and the protein is lactose permease. So, okay, there's three potential mark points there, but as I said, I think this question could come up in a longer form potentially. Okay, moving on to the last part of that question, it says lactose is a reducing sugar. Benedict's reagent can be used to detect the presence of lactose in the solution. A colorimeter can be used to measure the concentration of lactose. The colorimeter first needs to be calibrated. Describe how a method that uses Benedict's reagent and a colorimeter could be calibrated to measure the concentration of lactose in an unknown sample. So the question really tells you what it wants you to say. It says colorimeter, how to calibrate it, using Benedict's reagent, in an unknown sample. Okay, so those are key things. So let's talk about it. Okay, I've put some pictures over here so I can talk about it over here. So first of all, this is where you place the cuvette. So when you're calibrating, you would use a blank cuvette with some distilled water in it. Okay, blank cuvette with distilled water in it. So H2O in the cuvette, put it in there, and you press R, which gives it a kind of a reference value, and it will kind of set at 0. Now in the colorimeter, you do actually have to use a filter. So which filter to use? What you would actually be using a red filter when doing the Benedict's test. So yeah, you use a red filter when doing the Benedict's test, and that's just something you need to know. Okay, so then you do a range of Benedict's tests with known samples. So what you do is you get known samples with known concentrations of lactose, and you do the Benedict's test with these known samples, and you measure their absorbance at 635 nanometers, that's with the red filter, and you plot them like this. You do a line of best fit, or a calibration curve. Then you would repeat the process again using your unknown sample, the same Benedict's test, same concentrations of everything, and you'd get an absorbance reading. So your absorbance reading would be here, let's say, and then you just you go across on your graph, you'd hit your line of best fit, and then you go down, and then that would be your concentration of lactose for the unknown sample. Now, some of you might have been sort of thinking, oh, well, lactose wouldn't work because Benedict's solution only works with glucose. Well, lactose is a reducing sugar. It says it right at the top there. Lactose is a reducing sugar, so you don't have to do anything fancy. If it was a non-reducing sugar, then you'd have to do acid hydrolysis of the disaccharide first, which is HCl, and then what do? You HCl, you hydrolyze it, then you add sodium carbonate to neutralize it again. Okay, let's look at the mark scheme. We've got 0 the colorimeter set to 0 using blank. And I think for this, I would say in our syllabus, you'd say a blank cuvette using distilled water, a red filter, use known concentrations of lactose, or list of stated concentrations. You could talk about producing serial dilutions, potentially would have been worthy of a mark. Constructing the calibration curve is worthy of a mark. Test the unknown sample and use it, read from the graph to work out the concentration. Okay, so there's lots of things you could have said there to get the 4 marks. So I'll scroll down just a little bit. If you wanted to pause, you could look at the examiner's comments about this question. Students did find it tricky, basically. Pause there if you want to. All right, this question, 6 marker. There is definitely going to be some nucleotides stuff coming up this year. That's the year 12 content on sort of nucleotides, and I think that this stuff could come up in something like in this sort of way. So the question I put in was, DNA replication and transcription are two processes that occur in the nucleus of eukaryotic cells. Compare DNA replication and transcription by describing the similarities and differences between the two processes. So replication and transcription. Now, in any question that asks you to describe the similarities and differences, you know, pretty much half the marks are going to be for similarities, and half the marks are going to be differences. Okay, so you might even just, you know, in the margin, just kind of put those 2 words up, and then just try and like put some bullet points down. So I'm going to show you the 2 animations of these 2 processes, just to refresh your memory, and then I'll get into the mark scheme. So let's look at first of all, the animation for transcription. Okay, so transcription, first of all, we have the DNA here, and it's twisted and coiled up. So transcription, the first step is we have helicase enzymes and also gyrase enzymes that unwind and unzip the DNA, and they break the hydrogen bonds between the bases. Second of all, we have free nucleotides that sort of float in and line up next to the nucleotides on the DNA. So here are the free nucleotides floating in. And in this case, there'd be free RNA nucleotides containing the sugar ribose and the base uracil instead of deoxyribose and thymine. Okay, so the free nucleotides line up and they form hydrogen bonds through complementary base pairing. We have this enzyme called RNA Polymerase, I'll show it to you again, that moves along, and it doesn't actually form hydrogen bonds. What it does is it joins the nucleotides together down the backbone, so it forms phosphodiester bonds, the polymerase enzyme. As the mRNA is produced, it starts to slowly peel off the DNA sort of further back, and it will sort of wind up, and it will then exit the nucleus. Now, you know that because you studied module 6.1 that it may also be kind of processed and have introns removed by the spliceosome, if you watched my map on module 6.1. So that was the process of transcription, okay? Let's look at the process of DNA replication to compare it. Okay, so DNA replication. Again, we start off with the DNA helix twisted with hydrogen bonds down the middle. So the first step is using helicase enzymes and gyrase enzymes to unwind and unzip. There's a similarity. Then we have the sort of single strands of DNA, and we have free nucleotides sort of lining up opposite opposite the DNA strands. Okay, opposite the two DNA strands. And then we have a polymerase enzyme that moves along, joining the backbone. So there's a similarity. We have free nucleotides coming in, forming hydrogen bonds spontaneously, and a polymerase enzyme forming phosphodiester bonds up the backbone. Now, a slight difference here. DNA uses both strands to replicate. So there's free nucleotides that sort of fly in on the left strand and the right side. But in DNA replication, there's this weird thing where one strand is synthesized continuously and the other is synthesized discontinuously in fragments. Okay, so let's just kind of watch the whole process kind of go sort of as it kind of moves along. So this is a kind of nice little summary of the process. The helicase enzyme unzips and unwinds. We have polymerase enzymes joining the backbone, and both strands are being formed. So both strands are being used to produce new DNA. Okay, so eventually once the whole DNA has been replicated, the kind of strands separate, and then we've got two DNA molecules, not strands, two DNA molecules per chromosome joined in the middle of the centromere. So there are similarities there and there are differences. Now, it may be difficult to kind of tease out from your kind of knowledge of the processes the actual similarities and differences. So I've kind of just got two sort of summary pictures here. So similarity 1, gyrase and helicase enzymes. Similarity 2, free nucleotides lining up complementary base pairing hydrogen bonds. There's a few in there. Similarity 3, polymerase enzymes join up the backbone by forming phosphodiester bonds. But there are differences. One produces messenger RNA, one produces DNA. One uses free RNA nucleotides, one uses free DNA nucleotides. One uses RNA polymerase enzyme, one uses DNA polymerase enzyme. One uses only one strand, the template strand, to produce RNA, and one uses both strands to do semi-conservative replication of DNA. So those are some differences. Let's look at the mark scheme, because I probably missed out some more. And they're all here. Okay, so I'm just going to talk you through a little bit. I won't go through each individual similarity and difference, because I think we've kind of done that enough, but there's the similarities, all these points here. And there's the differences, and it's all these points down here. Okay. In terms of the kind of levels of response, 1 or 2 is a description of similarities or differences. So you didn't talk about both. You just talked about maybe 2 similarities, but you didn't talk about both. 3 or 4 is some similarities and differences with only minor errors. So maybe you've talked about like 2 similarities and 1 difference, and maybe your use of scientific language isn't perfect. And to get 5 or 6, you really need to talk about most aspects of the 2 processes with no significant errors. So you really want to kind of give, really 3 similarities and 3 differences is what I would sort of say is most with no errors. Okay, so pause there if you want to have a further look. Okay, moving on. I threw in a little multiple choice question here just because I wanted to get something on this topic of kind of basically white blood cells. So which one is a neutrophil? Out of this, the neutrophil is going to be B. So you need to remember that neutrophils have a lobed nucleus. The reason is it helps them kind of squeeze through small gaps out of capillaries into the tissues when they need to. And A is going to be a monocyte, which is a large white blood cell. Again, it's a phagocyte, and monocytes become macrophages. So sometimes they use interchangeably monocyte / macrophage, but technically speaking, monocytes become macrophages once they migrate out into the tissues. D are red blood cells. C is a lymphocyte. This is just good recap for you, because lymphocytes, the nucleus takes up the majority of the cytoplasm, okay? And this here, that would be a platelet. Okay, so yeah, that's the answer. B is neutrophil. Okay, moving on. Question 9. Bread again. Put quite a few questions on bread in this paper, apparently. So bread contains a mixture of polypeptides known as gluten, gliadins and glutenins, like we saw before. The table below shows statements about the structure of gluten polypeptides. So this is just a question on levels of protein structure. So what are the levels of protein structure? Short alpha helical sections are present. So alpha helix is secondary. Intermolecular bonds form between glutenin and gliadin polypeptides. This is quaternary, because remember, quaternary structure is anything to do with multiple polypeptides joining together, or if we have a polypeptide and then a prosthetic group is also coming in, for example, in the protein myoglobin or hemoglobin. 45% of the amino acids in gliadins are glutamine. This is talking about primary structure, because primary structure is just the sequence of amino acids. And this one is tertiary structure, because it's talking about hydrophobic and hydrophilic interactions and the way that causes one polypeptide to coil up into a shape. Okay, so here's a little picture just to remind you of those levels of protein structure. There you go. Make sure you know this stuff really well. It's in molecules and it is in advanced information. It could come up. Okay, moving on. We're still talking about the proteins, but now we're talking about the person's immune reaction to these gliadins in a person's digestive system. It says, the immune system produces antibodies that bind to part of the gliadin polypeptides, which cause inflammation. Some people who stop eating foods that contain gluten still occasionally experience the symptoms of celiac disease. Why is this? So celiacs are people who have an autoimmune, sorry, it's not autoimmune, just an immune response to the polypeptides. It's not autoimmune, I take that back. It's kind of like an overreaction, I would say. It's not auto, it's an overreaction to a really harmless polypeptide, but then it causes a lot of inflammation. So what can you conclude about the structure of the antibody that causes celiac disease and what the antibody binds to when producing the symptoms of the celiac disease? Well, basically, we know that the antibody normally binds to the part of the gliadin polypeptide, but it can also bind to other things, because even if you stop eating gliadin, bread basically, sometimes the antibody binds to other stuff. So the antibody must be able to bind to similar proteins, or it might bind to, or the antibody itself might be not very specific. It might bind to multiple things. So those are kind of what you can get marks for. I'll show you the mark scheme for the more specific answers. So you could have taken it a couple of different ways. Either you could have said other foods have the same antigen. You need the word antigen underlined there. Or you could have said the antibody is not specific. The binding site is not specific. Now, I'm not even sure if I would have got the marks to this. I think I spoke too casually. You really got to use your language correctly. So antigen is the thing the antibody binds to. Use that word. It's underlined here. And you could have got marks for saying that the antigen is a short sequence of amino acids. So maybe common to more than one polypeptide. Let's say one protein might equal, something like 1,000 amino acids, roughly, as a ballpark figure. And maybe the antigen is only like, 7 amino acids out of that 1,000, just a little tiny pocket. So it might be that multiple proteins have those same 7 amino acids kind of in one little spot of the protein. It might be common to more than one polypeptide. And what about the antibody? How does it work? How does it cause inflammation? Here's a little picture of how an antibody causes inflammation. So it can bind to a mast cell. Mast cells release histamine, which causes inflammation. This is on the disease topic. Remember, histamine, how does it cause inflammation? One of the things it does is it makes capillaries more leaky, so more tissue fluid is produced, which causes the area to kind of swell up. So that's just something to remind you about. Histamine is a signaling molecule that causes inflammation. That's why we take antihistamines when we have hay fever, because hay fever is, again, antibodies in the body kind of overreacting to something which is fairly harmless. Pollen, I get hay fever against like tree pollen, and I take an antihistamine to lessen my symptoms. Okay, moving on to question 10. This question is about some areas of heathland. So on a biology field trip, some students carry out a survey of butterfly species in 2 areas of heathland. One part of the heathland was regularly used by walkers, while the other had been deliberately fenced off by the National Park Authority in an attempt to promote biodiversity. Okay, so area 1 is accessible and area 2 is fenced off. Right, so how do they actually do this survey? So the survey was done on 2 different mornings in June. Students walked along a transect in each area 4 times. Okay, so 2 different mornings in June, transect 4 times at 30-minute intervals. So I guess that's taken like 2 hours. Yeah, that makes sense. And they recorded every butterfly sighting. The aim of the survey was to compare the biodiversity of the butterfly species in 2 areas. Suggest how the procedure could be improved so that a valid comparison could be made. Well, this is going to give you some information, but it's not going to be great. First of all, it's only June, okay? So we're just doing it 1 morning in June. Why not do it June, July, August, and then October, November, December? Do it in multiple months, do it at least, in 4 different months, in the different seasons, perhaps. So do it at different times of the year. There's only 1 transect being used in each area. Yes, we're walking along it 4 times, but only 1 transect in each area. We can do better than that. Maybe we should have multiple transects, 3 or 4 transects in each area. And the other thing is we're just recording sightings of butterflies. It's not brilliant. So maybe we might either misidentify butterflies, or potentially we could do some sort of catch and release sampling to get a better estimate of the population, as opposed to just, oh, there's a butterfly, I'm going to write it down. So there's probably more things you could have said. Certainly there are. Here they are. So idea of more transects, different times of day. Yeah, I only said the morning. It was the morning. Or times of year. It's actually the same mark point. So we talked about that. More transects, different times of day or times of year, different weather conditions, capturing individuals. So like a butterfly net or a sweep net. Use of a method to correctly identify the species. So this would be a key. Okay, a key is something like this, or an identification card, and that can kind of help you make the right call when you see something like that. Okay, here are the students' results. Area 1 and Area 2. We're going to need to use these later on, I can see. Identify the area of the higher species richness. So species richness is about the number of species. So in Area 1, we've got this. We've got 1. We've got greyling, heath, gatekeeper, hair streak. We haven't got this one. We've got this one. So in Area 1, we've got a species richness of 1, 2, 3, 4, 5 species. So SR for Area 1 is 5. And in Area 2, we've actually got a higher species richness, haven't we? We've got all of the species present, so we've got a 6. So the area is 2. There are a greater number of species present. You might go on to say the silver-studded blue butterfly is not present in Area 1. What about species evenness? Species evenness is to do with an even distribution of kind of organisms. So we're looking down the numbers and trying to sort of work out which one's more even. Now, I think it's this one. Oh, sorry, hang on a minute, this one. But let's do the maths to double check. So the evenness can be measured as the range. So from here, we've got from 2 to 11, which is a range of 9. And here we've got from 0 to 16, which is a range of 16. So there's a greater species evenness in 2, but then the justification is something like the range of the number of individuals is lower. Let's look at the mark scheme to make sure that we use the, say it the right way. it has more species. That's for the richness one. And for this one, it's the idea that the range of n is smaller. Okay, if you just used figures, you could have got that, but you might have said the range of the number of individuals is smaller, would be a good way of saying it. Okay, so the Simpson's diversity index question, if you got the right answer, then you don't need to stay with me and kind of work it all out. The answer here is 0.7976. And if you got that right answer, you get 4 marks. Okay, so if you didn't get that, let's go through it together and let's see how you get the right numbers. So remember, Simpson's diversity index is this formula. You do get given it. It is 1 minus, let's just pinch in and really focus on what all these things mean. 1 minus, now this big thing means the sum of. So it's the sum of n, which is the number of individuals, that's small n, divided by big N, which is the total number of individuals, squared. Okay, so you've got to sum this up. So we need to use the table. So down here, we'll work out what big N is. And we're working it out for number 2. So let me just rub this out here and leave some space. Okay, so first thing, we've got to add up the column, which we see here, to work out the total number of all the species in that area. So we've got 40 individuals is big N. Okay, big N equals 40. And then we should work out in this column, little n divided by big N. So it's this number divided by 40. So let's do that. Okay, so that's what you should have for your n divided by n. And then you've got to square each number. So let's do that. Okay, that's what you get when you square them all. And then we need to add all these up, because remember, we're talking about the sum of n over n all squared. So we're going to total these up, and then we get this number, which equals 0.2025. And then finally, to work out our actual value for Simpson's diversity, we need to do 1 minus this number. So 1, take away that, equals 0.7975. Great. All right, here's a 6 marker. So the student concluded that fencing off the area of heathland has increased the biodiversity of butterflies. Evaluate the validity of the student's conclusion using all the information you've been given, including table 2.1. Okay, so I guess the key phrase, well, we evaluate and all of the information. Those are 2 phrases that jump out. So evaluate means plus and minus. We want to have kind of be thinking about maybe like 2 or 3 things to say for positive and 3 things to say for negative. And all of the information, well, what have we got? We've got the method. They told us about the method. And we've also got the sort of the data. So maybe that's a way of planning it, okay? We want to say some positive stuff about the method or the data and some negative stuff. Maybe we want to try and break it down like this and kind of do a little grid. So what can we say about the method? I mean, not a lot great about it, but we can say that maybe you might say transect gets sample of species present or something. And to be honest, there's probably not marks for this, but just to make sure that we get everything. What about the negatives of the method? Well, we don't know. 1 time a year. Maybe weather dependent. Only 1 transect was done in each place. And also, we actually have no idea, this is important, no idea what the heathland was like before it was fenced off, of the change. We have no idea of the change because we don't know the starting kind of level of biodiversity before the heathland was fenced off. And we also don't know how long it was fenced off. Has it been fenced off for a week? And would we really expect that to improve the biodiversity? Or has it been fenced off for 10 years? In which case, maybe it has had an effect. So that's kind of stuff about the method. But what about the data? Let's go and look at the data. So the fenced off heathland, double just checking, it is number 2, isn't it? Yeah, area 2 is the fenced off heathland. So what about the fenced off heathland? It does have a higher species richness. It does have a higher species evenness. And it does have a higher index of biodiversity, which we calculate as 0.7975. This just seems to have disappeared from my map up here, from my run through up here. So those are all things you can say. Let's look at the mark scheme to drill down in everything you needed to say. Okay, first of all, things that you could have said to support the conclusion. These are the positive things that you could have said. Okay, so we've got the fact that Simpson's index of biodiversity is higher. Change color, sorry, I don't like that one. We've got, you could actually get a mark just for saying this. High Simpson's index means high biodiversity. Greater species richness and greater species evenness. You might have also been able to say just that the silver-studded blue only occurs in the fenced area. So those are positives, and we were looking for you maybe to say 3 of those things, 2 or 3 of those things. What about kind of negatives? And this is negatives to do with the data. I didn't actually talk about this a lot, so this is something that I missed out. The difference in the Simpson's index is not big, okay? So you could have worked out the percentage difference between the two. One is like 7.97 whatever, and the other one was like 7.2 or something, so not that big. The range in the number of individuals is greater in area 1 than area 2. I don't really know what that means. I don't really like that point, because that means the species evenness is bigger in area 2, which is, yeah, I don't like that point. There's a higher number of individuals of large heath and gatekeeper butterflies sighted in area 1. So someone might go to area 1 and say, oh, look at all these gatekeeper butterflies. There's loads of them. This place has got high biodiversity. Well, actually no, but you can see how that might go against the conclusion. And this is what we said, that we don't know the length of time an area has been fenced off. Limitations of the method. Samples taken on only 2 days, taken in only 1 season. And then this more stuff here. No method to prevent recounting, and observation at a distance might have led to misidentification. So how did you get marks for this? What's the kind of breakdown? Level 1 or 2 is basically some discussion, but you've either talked about the validity of the method or the data. You didn't talk about the method and the data. 3 or 4 marks, you've talked about the method and or the data, and there's some judgment. So there is some holistic judgment of the information. So overall, I think that the conclusion is valid or invalid. And for the 5 or 6 marks going up here, what do we need? Full and detailed evaluation. We need to talk about the method, we need to talk about the data, and there needs to be a holistic judgment. Using evidence for and against the claim. So full and detailed, I interpret to mean maybe 2 or 3 positives and 2 or 3 negatives. 1 or 2 positives about the data, 1 or 2, let's say 1 positive about the data, 1 positive about the method, 1 negative about the data, 1 negative about the method, and then a few extras. That would kind of get there with a holistic judgment at the end. Okay, so that's kind of what you're looking for. Okay, next one, nice easy one here. Describe how to do the emulsion test for lipids and how a positive result would be identified. Well, here's a picture of it. Basically, you take your sample and you add ethanol. The ethanol, the reason behind it is the ethanol dissolves the fat. Fat is ethanol soluble in pure ethanol. And then you might filter it to take the ethanol, which has been filtered. And then into that ethanol, you add a few drops of water. Now, when that water goes in, the fat doesn't like the water, and it comes out of solution as a cloudy emulsion. So that's what we're looking for, the cloudy emulsion. The mark scheme. is here. Mix with or add ethanol or alcohol and then water, and then it goes cloudy. So you have to have ethanol and water and then goes cloudy. Except a milky white emulsion. Do not credit precipitate, because precipitate is something that sinks to the bottom, and actually the milky emulsion, if you leave it, will actually float to the top, so it's the other way. Okay. Next one. Phospholipid molecules contain fatty acids. Explain how the fatty acids in phospholipids allow the formation of membranes. Let's just go straight to the mark scheme for this one. I think it should be fairly should be kind of an easy question. What you needed to say was that the fatty acids are hydrophobic, which means that they cluster together. The phospholipid bilayer is formed, and the fatty acid tails are pointing inwards. You could also have said that the phosphate groups are sort of facing outwards, forming hydrogen bonds with water on either side. In longer, sometimes I've seen this question as a 3-marker or even a 4-marker, which requires you to just give a bit more sort of detail. All right, this question is about the control of the heart cycle. Sorry, not the control of the heart cycle. It's about pressure changes in the heart. So first of all, the question asks you to draw a line, let's draw it in blue, that shows the pressure changes in the right ventricle. So you need to remember that the right ventricle pumps with a lower force than the left ventricle and achieves a lower pressure. So it's going to mirror pretty much the left ventricle, but it's not going to go as high. So it's going to go something like this. And the timing of everything is going to be the same, but it's just going to be kind of lower, something like that. And actually the marks you get, one for the shape being the same and one for it being a lower peak is kind of what you're looking for. So the next question was a 6-marker, and it says, describe the events taking place at the points marked W, X, Y, and Z, and explain how these events are related to changes in pressure. Changes in pressure. So this was a bit of a tricky question. I'm just going to get rid of this line now, just so I can go through it really clearly, because you needed to talk about what was happening to the heart muscle. Is it contracting? Is it relaxing? What phase are we at? And we also needed to talk about the valves. Are they opening or closing? And the biggest thing that we didn't really talk about was pressure in relative terms. You need to talk about what pressure is higher than what pressure. So let's first of all look here. So between this point to this point, or this region here, this is going to be, the yellow region is going to be atrial systole. So you need to say that. The atrial systole is occurring before point Y, and during this time, the pressure in the left atrium is above the pressure in the left ventricle. So I'll just write that on here. Left atrial pressure is above that of the left ventricle. But then we move into a new phase. Now, the pressure in the left ventricle shoots up enormously. We're kind of probably to around about here. It's shooting up enormously. So what's going on here? Well, this is now ventricular systole. And right at the beginning of ventricular systole, at point Y, this is the first thing you really need to talk about in detail, the pressure in the LV now becomes greater than the pressure in the L atrium. So the pressure in the left ventricle rises above the pressure in the left atrium, so therefore there's a valve that closes at Y, and it's the AV valve closes. Okay, you don't need to say bicuspid or tricuspid, it's just something to easily get wrong, so I wouldn't even try. Just atrioventricular valve closes. All right, so the pressure climbs and climbs and climbs. The left ventricle here, which I'm now going to kind of go over in blue, is going up, 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 up until the point Z. What happens at Z? We're still in ventricular systole here, but at Z, now we can say that the pressure in the left ventricle is now above the pressure in the aorta. So I'll do the aorta in green here, I guess. So now it's gone above that, is above the aorta, which means that the semilunar valve opens. Okay, SL valve opens. So it stays open whilst the pressure is higher in the left ventricle. Whilst the pressure in the left ventricle is higher, it stays open. But then eventually, as the let's highlight in a different color now. What color should we do? Let's do green. So now here we're in this region. The kind of pressure is decreasing. It's no longer peaking. So the pressure is decreasing. So really now the ventricles are relaxing. So this is all diastole over here. So this is diastole. The pressure in the ventricle is dropping, and once it drops below the pressure in the aorta, then the semilunar valve closes. SL valve closes, and the pressure in the ventricle keeps dropping, and then it crosses over again. Look, it's crossed over. Let's put this. Let's just fully colorify this. Because as you know, I like colour, so let's do the atrium pressure as in red here. So atrium is in red and the aorta is in green. So now at X, we could say that the left atrial pressure is again higher than the left ventricle pressure, which means that I need to make these colours more clear, don't I? That should be LV, blue, LA, that's LV. Yeah, let's do that. Okay, yeah. Oh no, this one should be blue here down here. Yeah, right, good stuff. All right, so now the left atrium is above the left ventricle, which means that the AV valve opens. Okay. So to get 6 marks in this question, you basically have to give all that information. You have to say what's happening with the heart. Is it contracting? Is it relaxing? What's happening with the pressure? Is the pressure higher or lower? And what's happening with the valve? Is it opening or closing? So to get the full 6 marks, the mark scheme says this. Okay, I'm just going to go straight for this description. 5 to 6 describes fully the behavior of the chambers and valves at W, X, Y, Z, and relates these behavior to changes in pressure. Changes in pressure at all of the points W to Z. So if you missed out any of those things, chambers, valves, W, X, Y, Z, you couldn't get 5, 6 marks. If you missed a couple of them, you would have got 3 or 4. And to get 1 or 2 marks, you talked about the chambers or the valves or the pressure, but you didn't do enough of those things. Okay, so we talked about all that in what I just said. Chambers is here. Valves is here and pressure is here. Okay, pause there if you'd like to have a bit more of a look. All right, final question. This is the kind of the only question I could really find on this stuff: apoptosis, mitosis, and meiosis. Which of these processes is important in determining the body plan of an organism? Well, apoptosis, yes, because remember, to make a hand, first you kind of like in the body, that is in the womb, the cells first kind of make a glove, and then the skin in between the fingers dies to make a hand. And also mitosis is making new cells, so that's important to build a body. You can control the rate of growth of cells. But meiosis, not really important during the development of a body plan. That's important in reproduction. So it's 1 and 2 only. So the answer is B. Okay, so I hope that paper was helpful. Here are once again the kind of questions there and kind of what part of the specification they relate to. So the questions are in a bit of a weird order, but if you want to use this, then please do. Look through the paper, see where you got marks, where you lost marks, and come up with an action plan for what you need to do to fix these areas, because these are areas that really, really might come up this year. Okay, I hope you found that useful, and I'll see you next time.